This is a production of Cornell University.
Thank you for filling in for Greg, because I know he's on sabbatical now, so it's nice to have a wonderful introduction. And thank you for having me here today to talk to you about my work, where I'm going to describe to you how we identified a tomato receptor that we call FLAGELLIN-SENSING 3, and it perceives the peptide FLAG228 and activates immune signaling in tomato. And so I just want to acknowledge this was a huge collaborative project, and I want to especially emphasize there were two postdocs in Greg's lab, Pat Boyle and Jill Rao, who were really helpful for this project. We couldn't have done any of the work without Susan Strickler, who's here today, thank you, who was a bioinformaticist who did a lot of data mining for us. And also we have two chemistry students, Josh Basili and Inish O'Doherty, who were really instrumental in some of our development of chemical probes that I'll talk to you about. And so without that, I'll start by reminding you of the overview of plant immunity. And so we have on the surface of a plant cell these pattern recognition receptors, or PRRs, that are responsible for perceiving pathogens. And I'm going to use the example of Pseudomonas, which is a bacterial pathogen, and it has a flagellin, and a part of that flagellin is this conserved region called microbe-associated molecular patterns, or MAMPs, that are perceived by these cell surface receptors and initiate intracellular signaling that include kinase cascades and that lead to translational reprogramming that leads to pattern-triggered immunity. And this pattern-triggered immunity is important for inhibiting the growth of pathogen. Now, of course, pathogens have been able to usurp this system through the use of effectors, specifically type 3 effectors, that are injected into the plant cell by the type 3 secretion system. And these effectors, once they're inside the cell, can then interfere with this pattern-triggered immunity. And in the case of AVR-PTOV and AVR-PTO, two effectors in Pseudomonas that we study in tomato, these specifically inhibit this pattern recognition receptor complex activity. And so thereby it inhibits pattern-triggered immunity. Now, of course, the plant has to fight back. And so in tomato, these two effectors are recognized by the resistance protein complex PTO-PTF, that then leads to effector-triggered immunity and programmed cell death that then stops the spread of pathogen. So today we're specifically focused on this very initial events of recognition that's happening on the plant cell surface. So in pattern-triggered immunity, we have these cell surface receptors, and these are transmembrane receptors, and the majority of them is the ones I'm showing today, all have intercellular protein kinases and these extracellular domains. So before, in the last slide, I had FLS2, which is probably the best characterized receptor in plants for immunity. And so FLS2 works with the co-receptor BAC1 and recognizes FLAG22, which is a region of flagellin. Now this is a fairly well-conserved receptor throughout plants, but there are several examples of receptors that are found in very specific plant groups or families. So in the case of the EFR receptor that recognizes ELF18, which is from the elongation factor TU of microbes, this is only found in Brassicaceae, so in Arabidopsis. Another example, so these are all examples of these leucine-rich repeat receptor-like kinases, which are indicated in these little green domains. We also have other ones, including the lysine domain-containing proteins, such as the SERPs, which recognize chitin. And I'll bring up that this was a new receptor that was just published yesterday in Plant Immunology. So this actually recognizes LPS, lipopolysaccharides, and its receptor is called LOR, and it's a lectin S domain protein, receptor-like kinase. And so what's key and what's sort of conserved in all of these is that downstream, since these are all protein kinases, they activate kinase cascades, they include transcriptional reprogramming, and this all leads to your basal defense. And so I want to talk a little bit more about these two peptides called FLAG22 that I already mentioned, and this other new peptide that was only identified about a year and a half ago from a tomato called FLAG228. And this is also from flagellin. And so I'm going to step back for a second and talk a little bit about human receptors. So it's been known that there's actually two flagellin receptors in mammalian systems. You have an extracellular receptor called TLR5 that recognizes one portion of flagellin. And you have an intracellular protein complex that recognizes a second portion of flagellin. structure and what the tomato receptors seem to recognize, we actually again have two portions, the FLAG22 region that's recognized by FLS2, and we have a second region, FLAG228, that's recognized by a previously unknown receptor, FLS3. And so one early hypothesis when we first identified this new MAMP was, well, this could actually, both of these peptides could be recognized by the same protein. So in other words, the tomato FLS2 could just be modified in such a way as to recognize both. So we tested the hypothesis pretty early on, and what we could find was, in fact, when you silence FLS2 in tomato and you treat with FLAG22, you see a reduced response to this peptide. But there's no difference when you treat with the FLAG228 peptide. So this seemed to provide evidence that, in fact, we have two different receptors in tomato that are recognizing the same protein, but just different portions of the protein. So just to give you some evidence that FLAG228 actually is functioning as a MAMP, so that it's actually inducing transcriptional change in the plants. So Ernan Rossley in our lab, who actually gave a talk, I think, back in the fall, he did a lot of experiments working with MAMP inducers in tomato and looking at the transcriptome. And so I just want to acknowledge Jay Worley actually did a reanalysis of some of this data, and that's what I'm showing you today. What he did is that what we see is that looking at the number of genes induced in tomato upon treatment with FLAG228, we have more than 2,200. But if we look and divide further treatments, such as when we treat with Pseudomonas fluorescens, which is a PTI inducer, 86% of the genes that are transcriptionally upregulated are overlapping with the genes that are upregulated with FLAG228. So this sort of suggests that one of the major PAMPs in fluorescence is probably flagellin, and again, FLAG228 treatment is giving us that same response. When we treat with a disarmed strain of Pseudomonas stringi pathobar tomato TC3000, we see that 90% of the genes that are upregulated are overlapping again with the FLAG228 induced genes. And this percentage goes down in the fully virulent DC3000 because that, as I showed you previously, is sort of suppressing the PTI induction. And then if we have a strain that doesn't deliver effectors and doesn't have a flagellin, it's pretty much escaping recognition. We're not really getting an induction of PTI-induced genes. So when we looked at this evidence, this gave us some confidence that FLAG228 treatment was inducing a response in tomato. And so we were interested in maybe figuring out what was going on, and I happened to be involved in a project where we were screening tomato heirloom cultivars to see if they had variation in their response to different MAMPs. So we had included FLAG22 and another peptide CSP22, but I'm showing you here the data for FLAG228 treatment. And what I want to emphasize here is we saw that there was one line, yellow pair, that was tested that seemed to have really, really reduced responses to FLAG228 treatment. And so I wondered, was this an anomaly? What was going on? And could we actually find other cultivars that were in fact having a differential response? And so I went through and screened about 200 different tomato cultivars and also wild species of tomato. And what I could find was actually several cultivars and a few wild species accessions that had this reduced response to FLAG228 treatment. And so again, we have this yellow pear tomato. We have two Pimpinella folium accessions, so this is one of the closest relatives to tomato, but it's a wild species. And then we had all of these different tomato cultivars that all showed this extremely reduced response. And in fact, when you look at the response, there really isn't anything there, that's just background. And so we were very excited and decided this really set us up for a really nice genetics approach, where I used the yellow pear cultivar, the non-responder, as one of my parents, and the Solanum pimpinellifolium LA1589 as my other parent. What was really nice is we actually have genomes of both parents, so this actually made the genetics really, really nice later on. So what we have is we actually did a cross and looked at the phenotype of the F1s. And so what we see if we treat with FLAG22 for either yellow pear, the other parent, LA1589, or the F1 hybrid, they all respond equally well to FLAG22. But now when we treat with FLAG228, yellow pear is completely insensitive to the peptide, but the LA1589 and the F1 respond. So now this set us up to do a bulk segregation analysis. So I showed you the phenotype of the F1. When we went to the F2, we had a nice segregation of the phenotype where we had responsive plants and non-responsive plants. And so we pulled the DNA from the responsive plants. And the idea was that if we could go and sequence the genomes, we wanted to look specifically for regions of the genomes where we had SNPs that corresponded to only the yellow pair parent, but not to the other LA1589 parent. And again, because we had the genome sequences for both of these, this actually ended up being fairly straightforward. And so this is where Susie comes in. So we developed this analysis pipeline. I just want to walk you through it. So we, again, grouped the susceptible plants, and then we sent the DNA off for next generation Illumina sequencing. And then Susie did sequence processing of that data where she trimmed the reads and filtered, and then she mapped those reads onto her yellow pair genome so that she could call those SNPs. And then what she ended up doing is looking for regions where the Solanum pimpinellifolium specific SNPs were located. And then that allowed us to find some candidates. So I'm going to show you an example of what this output data looks like. And so for 11 of the 12 chromosomes, this is generally what the data look like. We're looking at the percentage of LA1589 SNPs. And so if we see no linkage to our trait of interest, we should see 50%. And that's generally what you see. Obviously, you have a little bit of wiggling in the data, but what we see is generally a 50% SNP ratio. And so, again, about 11 of the 12 chromosomes look like this. One chromosome, instead, had a suppressed region where we had very, very few LA1589 SNPs. So it's right in here. And this corresponded to about a 2.5 megabase region. And when we looked at the genes there, we actually found 9 receptor-like kinase genes, which was our sort of candidate protein that we were expecting. And so looking at these 9 genes, I then designed primers looking at these different portions of the genome and did a little bit of fine mapping. And when we looked at the recombination, we saw a suppressed recombination around the region of this one gene, but not around these other genes. And so we very tentatively called this one receptor-like kinase gene our putative FLS3 gene. So tomato has been really nice in the last few years in that we have a lot of genetic resources that have been developed. So one of those resources that's been developed out of Michigan State is SolCap sequencing, and they took a whole bunch of different tomato cultivars and pimpinellifolium accessions, and they did SNP calling throughout the genomes. And so we could actually mine this data, and what we looked for was, Susie first did a whole genome analysis, so she took all the SNPs from the whole genome and developed this tree. And what we could do is we had 3 cultivars in that list that were non-responsive. So those 3 cultivars are not related if you look at the entire genome. So again, these are 3 cultivars that don't respond. If we just take that region of interest, around 10 megabases around that region, we now see that all of these cultivars now cluster together. So this again gave us some potential evidence that perhaps this region of the genome was of interest for us, and it could have like a common breeding history or a common lineage. We next then went to different Solanum species. So I pointed out tomato, of course, responds to FLAG228 treatment. We also know potato and pepper also respond, but petunia does not. And so now we have genome sequences of all of these. So we can actually go into the genomes and ask, well, do they have this one candidate FLS3 gene? And really interestingly, we have the tomato FLS3 candidate, we have pepper, and we have potato. But if you look, petunia and actually benthamiana do not have a clear ortholog for this gene. So this again was some evidence suggesting that maybe that this was the gene and it was a newly arisen gene that was not present in, it came about after the divergence of petunia and nicotiana from the Solanum species. Okay, so this is a whole lot of evidence. So let's get back to the gene, what it is. So it has 2 exons. And what we did is we sequenced all of those different cultivars to see what was going on in this gene. And what we found is that in yellow pear, the 6 other tomato cultivars, and in one of the pimpinellifolium accessions, we have a 4-base pair deletion in the gene. And so actually the one base pair deletion is sufficient because that causes an aberrant stop codon. And so we would predict actually that in all of these cultivars and accessions, this is actually a null allele. And then we had one other that was very interesting. So in the pimpinellifolium LA373, we actually had what appeared to be a full-length encoding gene, but there were 4 non-synonymous SNPs. And so you can actually see, if you go back to the data, we see that this LA373 wasn't really a null. It seemed to have some response, but it was greatly reduced. So this was kind of another really interesting allele for us to study. But first, before all of this, we have to actually show what is the gene doing, what is it encoding. Not surprisingly, it's leucine-rich repeat receptor-like kinase. So it has 27 extracellular leucine-rich repeats, a transmembrane domain, and a kinase domain. And now when we go and actually look at what it looks like on the ectodomain, if you take the FLS2 BAC1 FLAG22 co-crystal structure and you put the FLS3 gene on it and thread it, it looks very similar. But if you look at the actual amino acid identity, it's only about 35%. So while structurally it looks similar, because again the leucine-rich repeat domains are pretty conserved as far as their structure, the actual amino acid conservation is fairly low. And now, finally, putting it into Planta. So what's nice is I can use those yellow pear tomatoes to do some work in tomato. And what I first did was express this FLS3 gene into yellow pear protoplasts to see if they're able to now signal. And so if I just express FLS3, we don't get any activation of MAP kinases. But if we treat with FLAG22 as our control, we do get activation. If we just treat with the peptide, we don't get activation either. It's only when we have the FLS3 plus the FLAG228 that we actually now get activation of MAP kinase signaling. And just as a control, if you remember this EFR and ELF18 pair, that we can put this in tomato and we also get signaling. So this was suggesting to us that the gene that we had was probably the right gene, but we wanted to do some further testing. So now we expressed FLS3 in Nicotiana benthamiana, which you remember from the tree I just showed did not have a clear FLS3 ortholog. And so if we put in FLS3 in the FLAG228, we get a nice response. And if we put in the EFR and its cognate ligand ELF18, we also get a response. But if you have the wrong receptor ligand pairs, we don't see any response. So it's not that just expression of FLS3 causes aberrancy. So we had some nice data that was suggesting that FLS3 could be the receptor, but we wanted to do some further studies. So going back to this model, what we had is, as I mentioned, we had this aberrant stop codon in one of our alleles that really didn't do us very much good. But we had this other nice allele that had 4 amino acid changes, 3 in leucine-rich repeat domains, and when we looked at that, they weren't really conserved residues. It didn't seem to be really big changes. But when we looked at this 4th one that's actually in the highly conserved P+1 loop, we're doing a threonine to proline change. And so I hypothesized it was probably this amino acid change that was causing this reduction in response phenotype that we were observing. And then one other one I want to mention is that in kinases they have a highly conserved ATP binding site that's important for the actual catalytic activity of the kinase. And so in this case it's at this lysine 877. So we also use this amino acid to probe the function of FLS3. And so now we started making mutations. So we're looking here at total ROS production. If I put in FLS3 into benthamiana and treat with FLAG228, we get response. If I make this ATP binding site mutant, we lose the response. And also if I make the threonine to proline substitution that I mentioned, again, we have a reduced response that's sort of similar to what we were seeing in the plant. And just to confirm that, what I actually did was cloned out the other allele from the LA373, which we call FLS3-2. And similar to what we were seeing from the actual plant, the pimpinellifolium, we see a reduction in response, but it's not no response. And in fact, if we take that allele and we mutate the proline back to threonine, we can restore wild-type function. So this told us a couple of things, but it also really implied that the kinase domain or kinase activity is probably important for the function of FLS3 signaling. so next we wanted to actually show that we're getting an induction of UTI. And so what we did for these experiments was we took benthammiana plants, we infiltrated the leaves with agrobacterium that expresses FLS3, and then came back later and treated with our peptide. So this should induce our immune response. We then came in 8 hours later and we challenged the plant with our DC3000 in an overlapping circle. And what this allows us to do is that we look for whether we have cell death in the overlap region. If we don't, it means that we didn't induce an immune response, and so the bacteria won. If we were able to induce immunity, then we don't see cell death in that spot, and that means that the plant won. So this allows us to really probe whether we are getting an immune response from this receptor and treatment or not. And so that's a horrible picture, I apologize. What you can sort of see here is that if we infiltrate with only YFP and do the treatment, you see that we actually have the cell death in the overlapping area the majority of the time. And this suggests, again, that we're not inducing a clear immune response in that region. If we take the inactive FLS3, the one that had the ATP binding site mutation, we again also see that we have cell death in that overlapping area. Thank you. But when we have the wild type FLS3, we don't see that we have any cell death occurring in that region. So that was suggesting, again, that we were getting an immune response that was being initiated when we had FLS3 plus the cognate ligand. These experiments were done with Diane Dunn in our lab. Okay, and last part, of course, is the effector story, because that's really what our lab focuses on. And so it's been known that the effectors ABRP2 and ABRP2B suppress FLS2 mediated signaling. And not too surprisingly, they also suppress FLS3 mediated signaling for both ABRP2 and for ABRP2B. And so this is something, of course, we're really interested in exploring in Greg's lab, whether this is going to be the same as what occurs for FLS2 or if it's actually different. And this is something I think Teeny and Simon are going to be working on shortly. And then we wanted to ask, is FLS3 signaling dependent on BAC1? And again, FLS2 signaling is, so we would expect that it would be, but we needed to do the experiments anyway. So if we use benthammiana and we either have a control silencing or a BAC1 silencing, in plants that are silenced for BAC1 and express FLS3, we have a reduced signaling. And it's comparable to what we see for EFR. So this was some preliminary evidence BAC1 was involved. But I was able to recently do in vivo studies looking at the existence of FLS3 and BAC1 in a complex using co-immunoprecipitation. And so if I have FLS3 and BAC1 expressed in the same leaf, if I immunoprecipitate pulling on the BAC1, I'm able to detect FLS3. But if I have YFP instead and pull on YFP, I don't pull down the FLS3. And so this was again suggesting that we were getting FLS3 and BAC1 to exist in a complex together. And I will say this was actually after treatment with the peptide. And so again, this is more something else we're very interested in studying as well, but needs more work. And so last thing that we needed to do in order to actually show that this is in fact the receptor, we had to demonstrate specific binding between the ligand and the receptor. And so we did this using some chemistry probes that were developed through collaboration with Frank Schroeder's lab, and specifically current postdoc Josh Briselli and a former postdoc Inish O'Doherty. And they came up with these very clever systems to put on two important chemical moieties. The first one is this diazurine photocrosslinker, and the second one is this alkyne chemistry group. And so we made two probes, our FLAG228 and our FLAG22, that had what we call as our handle. And what this allows us to do is that with the photodiazurine crosslinker, we can use UV light, and this will covalently attack and bond to whatever's close by. The alkyne handle allows us to do click chemistry, and what this allows us to do is specifically put on a tag. But it lets us put on a tag after we've done the experiment, so that the tag doesn't interfere with the binding of the probe to its, of the ligand to its receptor. And so we could show that putting on this little chemical group, which is actually about the 1.5 amino acids in size, it's not very big, it didn't affect the activity of the probes. And so now we wanted to show the binding. So I'll walk you through these experiments because they're a little tricky. We first express FLS3 GFP in our Benthamiana leaves. We then harvest microsomes and plasma membranes to have an enrichment of our protein and to get rid of a lot of the contaminating cytosolic proteins, as well as our chlorophyll, because that would soak up the UV radiation and prevent the downstream analysis. So we have our microsomes that have our FLS3 GFP. We add our probe and any competitors, and so if our peptide binds to the receptor, again, this photo-cross-linker is now in close proximity, so that we can hit it with UV radiation and it now photo-cross-links. So we now have a covalent linkage between our receptor and our ligand. Since we have a GFP tag, we can do immunoprecipitation to pull down this complex between the receptor and its ligand. And now with this click chemistry handle, we can add on any tag we want, but we instead were adding on biotin. So we can use a click chemistry reaction to add on biotin azide, and then allows us to do immunoblotting, looking for either the GFP or for the biotin. So we first could show that the probe chemistry was working. So when we treat with FLAG228 in the presence of FLS3, we get a biotin label, as you can see through the streptavidin HRP signal. And we have protein in both. So the FLAG22 is not just aberrantly binding. So it shows that our probe chemistry is specific and it's not just cross-linking everything. And we could also compete off the signal. So when we have our probe FLAG228 in the presence of excess unlabeled FLAG228, we lose our signal. And we don't with the FLAG22. So that would suggest again that we had a very specific receptor-ligand interaction between our FLAG228 and our FLS3, and that we couldn't get rid of this binding by just adding any old peptide to the experiment. And so, just sort of in summary, what we have now is we have identified FLS3, which is the receptor for FLAG228. We've shown that the signaling involves BAC1 as a co-receptor, and that activation of the signaling pathway leads to the activation of MAP kinase cascades as well as transcriptional reprogramming of the plant. We showed that the signaling involves the ROS production, and that the Pseudomonas syringae effectors AVR-PTO and AVR-PTO-B can come in and actually suppress the signal. And with that, which was very fast, I'm going to go ahead and just again acknowledge everyone in the group. So again, Pat did a lot of the cross-linking experiments. Zhulong was really instrumental in some of the genetics and also the cloning of the alleles. Susie did all the bioinformatics work, which was totally essential for this project, as you saw. Josh and Inish did the chemistry. We also worked with Joyce and one of her techs, Kate, with working with some cell cultures. And we also collaborated with Boris Benitzer and his student, Chris Clark, who were the ones who originally were finding the FLAG228 molecule. And with that, I'd be very glad to take your questions.
This has been a production of Cornell University, on the web at cornell.edu.